Welcome back to the deep dive. Today, we're going to be tackling the fascinating world of drug discovery.
Oh, very cool.
Specifically, that initial spark of an idea
Okay.
and the journey from, wouldn't it be cool if,
Right.
to actually pinpointing a target for a new medicine.
It's a bit like setting out on an expedition, you know?
Hmm.
You want to discover something valuable,
Right.
but first you have to choose the right terrain to explore.
Okay. I like where this is going.
Yeah.
So we're talking about the very early stages,
Uh-huh.
even before scientists start tinkering in the lab.
Exactly.
Okay.
Our source material, pharmaceutical product development,
Mhm.
in vivo correlation,
Right.
emphasizes the crucial role of unmet medical needs and deep biological insights in shaping these initial steps.
You've piqued my curiosity.
Good.
What kind of needs are we talking about, and how do they spark ideas for new drugs?
Well, imagine a disease with no effective treatments, or a treatment that's effective but comes with a laundry list of side effects. Oh, gosh.
Those are glaring needs that scream for innovation.
So it's like finding a problem that's begging for a solution.
Yeah.
A medical mystery waiting to be solved.
Precisely.
Okay.
Let's take digoxin,
Okay.
a heart failure medication. Scientists knew it was effective,
Right.
but also that its absorption varied wildly depending on the formulation.
Interesting.
That unmet need for consistent delivery-fueled research into how particle size affects digoxin's dissolution and bioavailability.
Fascinating. So recognizing those unmet needs helps set the stage for drug discovery.
Yes.
But how do scientists actually go from a broad need to a specific target for a new medicine?
Think of it like choosing the right lock to pick.
Okay.
First, you need to understand how the lock works, right?
Okay, I'm following.
Okay.
So in this case, the lock is the disease mechanism, and the key is the drug.
Brilliant analogy.
Thanks.
Scientists need to delve into the intricate biological pathways behind a disease.
Pinpointing the molecules or processes that are going haywire and contributing to the problem. That's how they find potential drug targets.
It sounds like detective work at the cellular level.
Yes.
Looking for those molecular culprits.
It absolutely is. Take the antifungal medication gruzofolvin. It was initially plagued by poor absorption.
Oh, wow.
By understanding that the drug itself wasn't the issue,
Okay.
but rather its low solubility,
Right.
scientists were able to create a micronized formulation that dramatically increased its effectiveness.
So it's not just about finding a target.
Right.
It's also about understanding the target's quirks and how a drug might interact with it.
Exactly. Once potential targets are identified,
Okay.
they need to be rigorously evaluated.
Okay.
Remember, not all targets are created equal.
So what makes a good drug target?
Yeah.
What are scientists looking for in this molecular lineup?
Think of it as a three-legged stool:
Okay.
druggability, safety, and efficacy.
Okay. Break that down for me. What does it mean for a target to be druggable?
A druggable target is one that can be effectively modulated by a drug molecule.
Okay.
Imagine trying to open a lock with a key that's the wrong shape. It just won't work.
Oh, I see.
Right.
So the drug needs to be able to fit the target and have a real impact on its activity.
Precisely. And of course, safety is paramount.
Right.
Interfering with the target shouldn't cause unacceptable side effects.
Yeah.
You don't want to fix one problem only to create a host of new ones.
Right. It's like making sure the key doesn't accidentally unlock a bunch of other doors and cause chaos.
Exactly.
And what about efficacy?
Efficacy means that hitting the target should have a real meaningful impact on the disease.
Okay.
The key needs to actually open the right door and lead to the desired outcome.
Finding a target that checks all those boxes, druggable, safe, and efficacious.
Yeah.
That's got to be like finding a needle in a haystack.
It's one of the biggest challenges in drug development, no question.
Right.
Scientists are constantly pushing the boundaries, using cutting-edge technologies and insights to sift through potential targets and identify the most promising candidates.
Wow.
Yeah.
So finding those ideal targets is like a high-stakes treasure hunt with scientific ingenuity as the map.
I like that analogy.
But let's dive a little deeper into this idea of druggability.
Okay.
What are some of the factors that determine whether a target can actually be modulated by a drug?
That's a great question.
Thanks.
One key factor is the target's structure. Think of a drug molecule like a puzzle piece.
Okay.
And the target like a puzzle board.
Right.
For the drug to work, it needs to fit snugly into a specific spot on the target.
So the target needs to have a binding site.
Yes.
A pocket or a groove where the drug can latch on.
Exactly.
Okay.
And that binding site needs to be accessible to the drug.
Right.
If it's buried deep inside the target molecule, the drug might not be able to reach it.
It's like trying to fit a key into a lock that's hidden behind a wall.
Right.
No matter how perfectly the key matches the lock, it's useless if you can't get to it.
That's a perfect analogy. Another important factor is the target's function.
Okay.
Ideally, you want to target a molecule that plays a key role in the disease process, something that's essential for the disease to thrive.
So it's like finding the weak link in the chain.
Yeah.
The Achilles heel of the disease.
Precisely.
Okay.
And you want to make sure that hitting that target will have a significant impact on the disease.
Right.
Ideally, stopping it in its tracks or at least slowing it down.
But how do scientists actually figure out which targets are worth pursuing?
Right.
It seems like a daunting task sifting through all those molecular suspects.
It is a challenge,
Yeah.
but thankfully, scientists have a growing arsenal of tools and technologies to help them.
Okay.
One powerful approach is high-throughput screening.
High-throughput screening, what's that?
Imagine a giant library filled with millions of potential drug compounds,
Oh, wow.
each one slightly different from the next. High-throughput screening allows scientists to test these compounds against a target.
Right.
In a very rapid and automated way.
So it's like speed dating for drugs,
Uh-huh. Yeah.
trying to find the perfect match for the target.
That's a fun way to think about it.
Yeah.
And once they've identified some promising hits,
Go.
they can then start to optimize those compounds.
Right.
Tweaking their structure to improve their binding affinity, their potency, and their overall drug-like properties.
It sounds like a process of refinement,
Yes.
starting with a rough diamond and carefully shaping it into a brilliant gem.
That's a beautiful analogy.
Okay.
And of course, throughout this entire process, safety is always top of mind.
Right.
Scientists are constantly evaluating the potential toxicity of these compounds, making sure they're not causing harm while trying to do good.
So it's a delicate balancing act, trying to maximize efficacy while minimizing risk.
Exactly.
Okay.
And it's a process that requires incredible precision ingenuity and a deep understanding of both biology and chemistry.
Okay, so we've talked about unmet medical needs,
Yeah.
the characteristics of good drug targets, and the tools scientists use to find and refine those targets.
Right.
But our source material also mentioned something called the Biopharmaceutics Classification System,
Right.
BCS.
Yes.
How does this fit into the picture?
The BCS is a brilliant system that helps us understand how a drug's properties influence its journey through the body.
Right.
Specifically, how well it gets absorbed from the gut into the bloodstream.
So it's like a roadmap for drug absorption, helping scientists predict how a drug will behave based on its inherent characteristics.
Exactly. The BCS classifies drugs into 4 categories based on their solubility and permeability.
Okay.
Solubility, as we've discussed, is how well a drug dissolves in fluids, and permeability is how easily it crosses biological membranes.
Right. Those are 2 key hurdles a drug needs to overcome to get into the system.
Absolutely. Class 1 drugs are the superstars. High solubility and high permeability. They dissolve easily and zip across membranes without a hitch.
So those are the drugs that have an easy time getting absorbed. No special tricks needed.
You got it.
Great.
Then we have class 2 drugs,
Okay.
which have low solubility but high permeability.
Right.
They can cross membranes readily,
Okay.
but their absorption is limited by how well they dissolve.
So it's like having a sports car with a flat tire.
Oh,
It has the potential for speed, but it needs a little help to get rolling.
that's a great analogy.
Thanks.
And this is where formulation strategies become crucial for class 2 drugs.
Okay.
Scientists can use techniques like particle size reduction or special excipients to boost their solubility and improve their absorption.
So it's like giving that sports car a new set of tires, allowing it to reach its full potential.
Precisely.
Okay.
Then we have class 3 drugs.
Okay.
High solubility but low permeability. They dissolve well, but they have a harder time crossing those biological barriers.
So it's like having a key that fits the lock but can't quite turn it. It needs an extra push to unlock the door.
Exactly.
Okay.
And in this case, scientists might explore strategies like permeation enhancers, substances that can temporarily increase the permeability of membranes,
Interesting.
giving those class 3 drugs a helping hand.
It's like lubricating the lock, making it easier for the key to turn.
Perfect analogy.
Okay.
And finally, we have the class 4 drugs.
Okay.
Low solubility and low permeability. They face an uphill battle on both fronts,
Right.
making them the most challenging to work with.
It's like having a rusty key that can't quite fit a stubborn lock.
Yeah.
You need a whole toolbox of tricks to get that door open.
That's a great way to visualize it.
Okay.
Developing effective formulations for Class 4 drugs often requires a combination of strategies.
Right.
And even then, achieving adequate absorption can be tricky.
So understanding the BCS classification
Mhm.
is like having a cheat sheet for drug absorption,
Yeah,
helping scientists anticipate potential hurdles and design strategies to overcome them.
absolutely.
Okay.
It's a powerful tool that helps guide the development process and ensures that scientists are choosing the right approaches for each drug,
Right.
ultimately increasing the chances of getting effective treatments to patients.
Okay, so we've explored how unmet needs spark drug discovery,
Right.
the intricate process of target selection,
Mm-hmm.
and the importance of understanding drug properties through the BCS.
Yes.
But our journey isn't over yet.
Yeah.
There's one more fascinating area we need to delve into. In vitro. In vivo correlation, or IVIVC.
IVIVC, the bridge between the lab bench and the human body. It's
Mhm.
like having a crystal ball.
Okay.
That helps us predict how a drug will behave in the real world based on its performance in laboratory tests.
That sounds incredibly powerful.
Mm.
But how does it actually work? How can we connect the dots between what happens in a test tube and what happens in a living, breathing person?
It all starts with dissolution testing,
Okay.
which we've touched upon already.
Right.
Remember, dissolution is the process of a drug dissolving in a fluid,
Right.
like those found in our gastrointestinal tract.
Okay.
It's a crucial step for absorption,
Hmm.
as a drug needs to dissolve before it can enter the bloodstream.
Right. It's like making a cup of tea.
Uh-huh.
The tea leaves need to infuse into the hot water before you can enjoy the flavor.
Exactly.
Yeah.
And just like you can control the strength of your tea by adjusting the brewing time,
Right.
scientists can assess the rate and extent of drug dissolution using carefully controlled laboratory tests.
So these tests help us understand how quickly and completely a drug dissolves outside of the body,
Yes,
giving us clues about how it might behave inside.
precisely.
Okay.
And here's where IVIVC comes in.
Okay.
By comparing dissolution data from these in vitro tests
Right.
with data from in vivo studies like clinical trials.
We can build a predictive model.
So it's like finding a pattern,
Yes.
a correlation between how a drug dissolves in the lab and how it gets absorbed in the human body.
You got it.
Okay,
And the stronger that correlation,
right.
the more confident we can be in predicting a drug's performance based on its dissolution profile. This has huge implications for drug development.
Okay, I'm seeing the potential. So how does IVIVC actually help scientists create better medicines?
In several ways. First, it can help optimize drug formulations.
Okay.
By understanding how dissolution influences absorption,
Right.
scientists can fine-tune the composition of a drug product to achieve the desired release profile.
So if a drug is dissolving too quickly or too slowly,
Yeah.
they can tweak the formulation to get it just right.
Exactly.
Cool.
IVIVC can also play a role in reducing the need for certain clinical trials.
Really?
If we have a strong correlation between in vitro and in vivo data,
Okay.
some bioequivalence studies might be waived. Bioequivalence studies, those are the ones that compare generic drugs to brand name drugs.
Yes,
To make sure they work the same way, right?
that's right.
Okay. And if we can demonstrate bioequivalence through dissolution testing alone,
Right.
it can save time and resources, ultimately getting those generic medications to patients faster.
That's a win for everyone. Less expensive medications and a faster path to market.
Exactly.
Are there any examples of how IVIVC has been used to improve real-world drugs?
Absolutely. Remember that heart failure medication, digoxin, we talked about earlier?
Uh-huh.
Its bioavailability can vary significantly depending on how quickly it dissolves.
Right.
By establishing an IVIVC model,
Okay.
scientists were able to develop a formulation with more consistent dissolution,
Mm-hmm.
leading to more predictable therapeutic effects.
So IVIVC helped create a more reliable and effective treatment for heart failure patients.
Yes. That's incredible.
It's a testament to the power of understanding drug properties and their impact on the human body.
Wow.
And, as we wrap up this deep dive,
Yeah. I'm struck by the intricate web of knowledge that underpins drug development.
I know what you mean. We've journeyed from unmet needs to target selection,
Right.
from the complexities of drug properties to the predictive power of IVIVC.
Uh-huh.
It's a remarkable process that blends scientific ingenuity with a profound commitment to improving human health.
It truly is.
Wow.
And at the heart of it all is the desire to alleviate suffering to extend lives
Mm.
and to empower people to live healthier and more fulfilling lives.
So the next time you take a medication, remember the incredible journey it took to get there.
Yes.
From the initial spark of an idea to the rigorous testing and refinement, it's a testament to the boundless potential of human curiosity and the unwavering pursuit of a healthier world.
Absolutely.
Thanks for joining us on this deep dive.